In the previous video, we ended on page 71 where we talked about marasmus and kwashiorkor. In this video, we are going to talk about alcohol metabolism, and then we start from there until where we reach, then we pause, and then we continue the next video from wherever we are ending this one. If you are a returning subscriber, thanks for coming back to the channel. Comment so that I know that you are appreciating the videos, and also just give us some thumbs up over there, and also subscribe. If you are new on this channel, you are welcome to join us so that we review this first aid together. Yes, there's always something to be learned in a different way, different perspective, and then I hope this video helps someone who is watching it. So this session talks about ethanol metabolism. So that's what the page is about. We'll continue from here until wherever we reach in according to the pages of the first aid. So in alcohol metabolism, this ethanol is the same as alcohol, the alcohol that we drink. Okay, so when we drink it and it goes into our GI tract, the GI is able to break some part down. The liver is also able to break it down. So we have the enzyme that breaks it down. First of all, you have to know that this alcohol, when we drink it and it reaches the GI tract, alcohol dehydrogenase is the enzyme that is going to break it down. It will break it down into acetaldehyde. Then acetaldehyde will be broken down into acetate by the enzyme acetaldehyde dehydrogenase. Why do we say dehydrogenase? Dehydrogen, okay? That's how the enzyme was named, dehydrogen, remove hydrogen. So it's removing hydrogen from alcohol. And I told you that the alcohol, for instance, its functional group is OH. So it's just a chain of what? Carbon with OH on it, alcohol, or the OH. So we are just removing, when we split some of the hydrogens out, and then some of the carbons out, it means that we are breaking the alcohol down. So NAD, which is vitamin B3, is the one is the vitamin that will accept the hydrogen. So we remove this hydrogen here from the alcohol, and then that means that when we add the hydrogen to we add the hydrogen to NAD plus, it turns into NADH. Then this alcohol is then what? Oxidized to this, because anytime we remove hydrogen from something, it gains a plus, or it's being oxidized. But then when we add hydrogen, we are reducing it. So here, this NAD plus is being reduced to NADH, but ethanol is being oxidized to acetaldehyde, and this occurs in the cytosol. Take note of that. Occurs in the cytosol of the cell. So whether it's the enterocyte, it's the same thing. Whether it's in the hepatocyte, it's gonna be the same thing. So in the first place that it takes place is the GI tract. Next will be in the liver. Okay, and it's the same reaction. So you have to know that more of the alcohol dehydrogenase and then the acetaldehyde dehydrogenase are in the liver compared to the GI tract. Also remember that women have less of the alcohol dehydrogenase and acetaldehyde dehydrogenase. That is why if a woman takes a little drop of alcohol, they will get more drunk than men, because men have a lot of this alcohol dehydrogenase that will break down the alcohol, but women have just a little. So if they drink, they will get more drunk than men. So this is how alcohol is broken down into in the body, into the simplest form. Now you have to know that this thing over here, the end product here, they are usually toxic, but the most toxic one is this acetaldehyde. It can be toxic to the cell, so we must continue to break it down into acetate. It's toxic in that they say it burns like fire. Yeah, so that's how I remember that it burns like this. Acetaldehyde can burn like fire. The things that we have to memorize here or know for the step is that this drug is called formipixole. It's able to inhibit alcohol dehydrogenase so that it cannot break down ethanol. Not just ethanol, but then there are other things that look like ethanol. For example, we'll see them over here. Here, if somebody drinks methanol, ethylene glycol, they are toxic. And then when they are broken down, we'll get more of, for example, it will be for example, methanol will be broken down into formic acid. That formic acid is very toxic. We don't want it to form. We'll see it later. We don't want it to form. So if it forms, the cell will be damaged. So we want to rather inhibit the alcohol dehydrogenase. And also in other questions, what they want you to know is that, for example, when somebody drinks methanol, you don't want them to, that is it antifreeze or something? When someone drinks antifreeze, you don't want them to convert the antifreeze into whatever, or acetaldehyde or formic acid, which are toxic to the body. So we rather give them alcohol. Okay, we give them ethanol rather, and then the ethanol will now be used, or in another way, I'll say the alcohol dehydrogenase will now work on the ethanol instead of working on the methanol, because the methanol will be more end product of the methanol will be more toxic. So it's just like a decoy. So we can use alcohol to treat methanol toxicity. We'll see that later. We'll see that later. So yeah, it's something that we have to take note of, and then they love to test on that. So if somebody has a certain toxicity, let's say methanol toxicity, and then they are giving the person alcohol, or someone drinks antifreeze and they are giving him alcohol to drink in the hospital, that will be the treatment, because we are trying to prevent this enzyme from converting it into its toxic form. Next is that if somebody is a chronic alcoholic, we have a certain drug called disulfiram. We can give it to the person, and then it will block acetaldehyde dehydrogenase. If it blocks acetaldehyde dehydrogenase, it means that acetaldehyde cannot be broken down. We'll have accumulation of this acetaldehyde, and then when acetaldehyde accumulates, it becomes very toxic. It becomes very, very toxic to the body. Let me pull this from here. I'm sorry. I think I clicked the wrong thing. Yeah, it should be this one. So when acetaldehyde accumulates in the body, it becomes very toxic. So this over here will inhibit. This is the drug that is given to alcoholics, chronic alcoholics. You want them to stop taking alcohol, so that when we give them disulfiram, okay, which is also known as antabuse, where is it? Antabuse. Not use this thing. I don't know. It's also known as antabuse. I can just type it in here. Yeah, here. So it's called antabuse if you want the name. Yeah, another name for disulfiram is antabuse, like that. Yeah, and then this antabuse over here is can make it bold like that. Good. So antabuse is another name for the for the disulfiram. So this antabuse or disulfiram is given to a chronic alcoholic so that it will block acetaldehyde dehydrogenase. When we block it, acetaldehyde will accumulate. This acetaldehyde will make the alcoholic vomit. Like, it will make them vomit so much that anytime they bring alcohol to their nose, they'll vomit. They'll vomit so badly that they wouldn't want to go close to alcohol again. So it's used to treat chronic alcoholism. Yeah, so we can see that in this pathway, we are making a lot of NADs. That's what we have to appreciate. We are making a lot of NADH over here, a lot of them. Therefore, what will happen? It means that anytime someone drinks alcohol, we'll make a lot of NADH. Let's see why this NADH is not a very good thing. You remember that in glycolysis, we said that we make a lot of NADH on the way. In TCA cycle as well, we make a lot of NADH. When we have a lot of NADH, what does that mean? It means that the cell has high energy because we can use these NADHs to make ATP. Is that correct? We can use them to make ATP, right? This thing. Sorry, I'm still new to this. Yeah, we can use them to make ATP. Is that correct? So if we can use them to make ATP, it means that anytime they are high, it means that we have a lot of ATP being made. So it means that when someone drinks a lot of alcohol, they will also have NADH, and then the body will perceive all these NADH that are made from the breakdown of alcohol as what? High energy. Therefore, it will slow down the TCA cycle. If it slows down the TCA cycle, because the TCA cycle is there to generate ATP, NADH, and then ATP, correct? So if you have excess NADH, which we are not using, then the body thinks that there is excess energy already. So we don't need to proceed with this TCA cycle. So we say that the TCA cycle will back up. Now, if it backs up, what will happen? It means that we'll have excess of what? Isocitrate. We'll have excess of citrate, that is not written here. We'll have excess of what? Acetyl-CoA, and everything will back up. Everything will back up. Therefore, the person cannot what? Break down glucose well through this pathway. Everything will back up. So then, the next thing I will see is that when everything backs up, acetyl-CoA, excess of it will be used to make what? Ketones, keto acids. And these keto acids, we know them as what? Beta hydroxybutyrate, acetoacetate, and acetone. But one thing that you should know is that acetone is the one that is breathed out. Like we breathe it out, so we cannot detect that in the blood. It's always breathed out, so it's the one that gives the smell. But then beta hydroxybutyrate and then acetoacetate can be detected in the blood. So take note of that high yield. You should know that. Next, we can have excess what? Pyruvate also, because pyruvate will also accumulate. If pyruvate accumulates, what will happen? Anytime pyruvate accumulates, you'll have what? It will be converted into lactic acid. Therefore, if somebody takes a lot of alcohol, and then they have a lot of NADH, these are the effects of excess NADH, or the effects of excess what? Alcohol intake. So excess alcohol intake will cause what? The ratio will go up. Increase NADH and NAD plus ratio. Increase means that there is more of the NADH. Increase. So if you are dividing the two, this math, increase, if you are dividing the two, if the upper one is higher, it means that the number would increase. For example, if this is 5 and this is 2, 5 over 2 will give us like maybe 2.5, right? That means that the number is increased. But then if the upper one is smaller, the ratio will be decreased. So increase, anytime someone drinks alcohol, it's an increased ratio of NADH to NAD plus that causes the problem. So that means another way you can say that excess NADH will be formed, and that will slow down our glycolysis. And when it slows down glycolysis, everything backs up. Then acetyl-CoA will be used to make ketones. So alcoholics can have ketones in their blood. They can also have lactic acidosis, right? And then this acetyl-CoA can also be used to make fatty acid, as we saw in the previous video. Make fatty acid and then make a lot of what? Lipids, and then those lipids, or in the form of triglycerides, will deposit in the liver and cause hepatic steatosis. Yeah. And later on we'll see how that histology looks like on the liver. We have to understand that. I think that should be in GI. If you want to see it in GI, should I search for it? Let's see. Yeah, that's how it will look like. Let's just continue. You can look at GI and then look at the image of pause the video, look at the image of hepatic steatosis, alcoholic hepatic, they call it alcoholic fatty liver, or that's hepatic steatosis. So that is one thing. Next, over here you can see that we said this we can also have conversion of glyceraldehyde 3-phosphate, or that's G3P, okay, or glyceraldehyde 3-phosphate, to DHAP, that's dihydroxyacetone phosphate, right? And then this will also be converted into glycerol 3-phosphate. Glycerol 3-phosphate can also be used to make triglycerides. So we'll have more hepatic steatosis over here. We can read here quickly. It's the same thing that I've talked about. So increase NAD to NAD plus, NADH to NAD plus ratio increases, inhibits TCA cycle. As we saw down here, it inhibits the cycle, and that means that it leads to increase in the acetyl CoA, as we saw here, because everything will back up. And then that will be used to make, acetyl CoA will be used to make ketones, and also be used to make fatty acids, causing hepatic steatosis. And also, these fatty acids, they are depositing in the liver. If you look at the histology, you'll see a lot of fat in it. And so we had already said that females have females have less alcohol dehydrogenase. So females are more susceptible than males to effect of alcohol due to decreased activity of the gastric alcohol dehydrogenase, and then decreased body mass, and also decreased percentage of water in their body weight. So these are the three reasons why females are more susceptible to all these problems. So they have decreased alcohol dehydrogenase. That is the main thing for me. Then they have their body mass also, that is less than the percent. They have less water in their body to dilute the alcohol. NAD plus is the limiting reagent. Right here you can see that. And also alcohol dehydrogenase, let me use the highlight here, because so alcohol dehydrogenase operates via the zero order kinetic. So zero order kinetic, you just have to know that this is how it looks like. They'll draw this, and then you always see something like this. To be like this, and the kinetic always look like this. To also look like this. So maybe you have V max here, and then you have Km over here. So this is how it looks like. So you see that as the substrate go increases, as the substrate increases, you'll see that the velocity has plateaued over here. So that is zero order kinetic. This is how the graph will always look like for zero order kinetic. So take note of that. When we come to that part of biochemistry, I will talk more about that. This also has to do with pattern recognition. You just have to know, when you see, you must just know it straight away. That's how NBM is. And you love to test that concept. Ethanol metabolism increases what? NADH/NAD plus ratio in the liver. That's what we said earlier, and it's the same thing over here that is said in the liver, right? Causing lactic acidosis, as we said already. Pyruvate to lactic acidosis when everything backs up. And then we also have fasting hypoglycemia due to decreased gluconeogenesis, because everything has stopped. So this one, oxaloacetate, for example here, oxaloacetate, instead of being converted into PEP, to be converted into malate, but we need the oxaloacetate to do gluconeogenesis. So like that. So they have fasting hypoglycemia. They cannot make they cannot use oxaloacetate to make how do we call it? They cannot use oxaloacetate to make PEP, because this process needs NAD, right? It needs NADH, because oxaloacetate needs to be converted to PEP. This one is what? 4 carbons. It has 4 carbons. 1, 2, 3, 4 carbons. But pyruvate, PEP, let me see the number of carbons it has. If I wrote it there, I think I didn't write it in this book, but it looks like it should have 2 carbons, or it should have 3 carbons, probably. It should have 3 carbons. Yeah, it should have 3 carbons here, right? And then this one has, pyruvate has 2 carbons, acetyl-CoA has 2 carbons, like that. So now this oxaloacetate should be converted into PEP ideally, but then we need the enzyme that it needs is enzyme 16, that's PEP carboxykinase. And then the cofactor there is the niacin. So if we are having excess of the niacin, excess of the NAD plus, it means that we have little of the NADH, but we need the NADH in this reaction here. So little NADH means that oxaloacetate cannot be converted into the PEP. If you have a good diagram, you will appreciate it. But if you have a very good diagram, let me see if we have it somewhere. It doesn't seem to be in the book, but then you have to know that over here we need NADH, NAD. Over here we need NAD plus here, right? NAD plus here, NAD plus. We need it badly at this place, but now we've converted all of them into NADH, so therefore oxaloacetate cannot be converted into PEP. Understand that very well. And so all the oxaloacetate will be converted back into malate, and then so we cannot have gluconeogenesis. This is how gluconeogenesis occurs, from oxaloacetate to PEP, then it continues to glucose. Okay, now the next thing is that we talked about ketoacidosis, that we already talked about. Ketoacidosis, and then we talk about all the 3 types of ketones. Do you remember them? All the ketones. We talk of what? Acetone, beta hydroxybutyrate, and acetoacetate. They are the 3 ketones. Which one can be detected in the blood? 2 of them, the acetoacetate and what? Beta hydroxybutyrate. Which one is which can be smelled in our breath? That is what? Acetone. That's why we call it the acetone breath. Okay. Ketoacidosis, so diversion of acetyl-CoA into ketogenesis, that over here, as we saw. We've talked about that already. Also hepatic cells, we talked about it already. So this stuff are not that hard. Let's look at the formipixole. The formipixole that we saw over here. Let's see what they talk about it. So formipixole blocks alcohol dehydrogenase, preferred antidote for overdose of methanol. You must know this. When someone drinks methanol, ethylene glycol, I think that is the antifreeze, the ethylene glycol, we give them formipixole. Formipixole will block the alcohol dehydrogenase so that it's not converted into acetaldehyde, because this acetaldehyde is very toxic. Also, ethylene glycol can be converted into other substances that can be toxic to the cell. So we don't want that to occur. So alcohol dehydrogenase has higher affinity for ethanol than the methanol, and then the ethylene glycol, leading to ethanol can be used as a competitive inhibitor of the alcohol dehydrogenase to treat methanol or ethylene glycol poisoning. So that's what I was saying, that if somebody, for example, has drunk methanol or ethylene glycol, which is antifreeze, those things will be converted, for example, into formic acid. That formic acid is very toxic. We don't want that to occur. But then at the end of the day, it is alcohol dehydrogenase that is converting both of them into the formic acid. So what do we want? We want to give something to serve as a decoy so that alcohol dehydrogenase will focus on that rather, so that we don't convert the ethylene glycol to its product, end product, but rather we are finding the work for the alcohol dehydrogenase. And then they are saying that it has more affinity for ethanol than the methanol and co. So it means that when you put ethylene glycol and ethanol into the body, and then you have alcohol dehydrogenase will rather go and convert the ethanol, and it will leave the ethylene glycol. So if somebody drinks ethylene glycol, repeat after me, if somebody drinks ethylene glycol or methanol, we give them formipixole so that it blocks alcohol dehydrogenase and prevents it from converting them into toxic substances. Very, very high yield very high yield. Please, very high yield. Remember the enzyme Formipixol or Pizol. Yeah. And this antaboost is also used to treat chronic alcoholism. Yeah, that is disulfiram. I love to call it antaboost. So it blocks acetaldehyde dehydrogenase and increases acetaldehyde, and leading to hangover symptoms and discouraging drinking. So they vomit a lot, they vomit badly. And then when they take that drug and then they bring alcohol even to their nose, they begin to throw up badly. So they will not even go close to alcohol again. And then you don't want to give that drug to someone who lives alone, because they are going to vomit bad to death. So you don't want to give it to them alone. It's also another point. Now, they have some enzyme terminologies here. You know, a lot of people have problem with this enzyme, but I think it's one of the cheapest things that like we can learn. So I'm going to teach you how they name the enzymes, and that will help you to understand biochemistry very well. Okay. An enzyme often is described by its function. So for example, if the enzyme is removing hydrogen from something, we call it a dehydrogenase. If it's adding hydrogen, we call it a hydrogenase, right? If it's removing carbon, we call it decarboxylase, right? If it's adding carbon, we call it carboxylase, right? Now, the two main ones that you want to know first are the phosphatase, right? And then the phosphorylase, these two. Phosphorylation means that addition of what? Phosphate to anything. Example, if I have AMP and I add a phosphate to it, it becomes what? ADP. Is that correct? AMP, that's adenosine monophosphate. It becomes adenosine diphosphate. It is called phosphorylation because I've added phosphate to it. That's a phosphorylase. Now, if we have a phosphatase, phosphatase is doing the opposite of a phosphorylase. So if you have something we call, if you have, for example, phosphatase, and then, for example, when we get to how do we call it, gluconeogenesis, we'll realize that the final enzyme is what? The phosphatase enzyme. That is is it, how do we even call it? I want the name for it so that I don't make a mistake for you guys. Okay. This is it here. So it's called glucose 6-phosphatase. Good. Von Gehrke disease. So that is glucose 6-phosphatase. That's how they call it. So glucose 6-phosphatase, okay, it means that it is removing a phosphate from the glucose so the glucose can leak out of the cell. But then when we add we have a phosphorylase, the phosphorylase will be able to what? Add the inorganic phosphate. But let's look at another one here. We have the kinase. So kinase, phosphatase are directly opposite. Phosphorylase is also working like a kinase. Kinases add phosphate also to some how do you call it? To a substrate. Okay, but then you see the difference here is that the kinases add normal phosphate. These ones add, the phosphorylase add inorganic phosphate. Okay, so let's take note of that. Don't get confused. If I stated it wrong, just listen again. Kinases add normal phosphate. Phosphatases remove it. That same normal phosphate. But phosphorylases work on inorganic phosphates. So we'll see those later. Inorganic phosphates mean that they have a charge on it, but these normal phosphates do not have any charge on them. So take note of that. So anywhere you see kinase, it means it was like, for example, phosphofructokinase 1. It's adding what? Phosphate to the fructose, right? Phosphofructokinase 2 is adding a phosphate again. So you will see that glucokinase, hexokinase, they're adding phosphate to the glucose. Is that correct? Yeah. We'll see all those things. Now, We have dehydrogenase, we talk about that. They remove what? Hydrogen, dehydrogenase. Hydroxylase, they add OH here. Right, we have tyrosine hydroxylase, we'll see it, hydroxylase, just adding OH is like a water molecule, right? And carboxylase, we talk about it transferring what? Carbon dioxide group, CO2 group. Like for example, it's a carbon, I just call it carbon group, and it's pyruvate carboxylase is one of them. Mutase is like, it's just changing the thing, like they look, it's like identical twins, like they look the same, but then there's a little difference. Carbon molecules are the same, hydrogen, the number of carbon molecules are the same, the number of hydrogen molecules are the same. But for example, you find metal-malonyl CoA mutase that converts metal-malonyl CoA to succinyl CoA in the TCA cycle. If you remember that, we talked about that in the previous video. That's in the common pathway, that the yellow part in the bottom that I talked about. That is very important. We also look at this one here, which is the synthesis, meaning that they are making something, they are synthesizing something. So we can understand that also. So the enzymes are really not difficult. Kinases and phosphatase are the most important that we'll be seeing most of the time. Kinase, phosphatase. Take note of that. The rate limiting enzymes in metabolic processes. So these ones, as we go through them, we will see them. We don't have to memorize all of them right now because we've not seen the pathway, so it will not make any sense to be learning all this rate limiting enzyme, because you have to reach the pathway before you understand what is going on. Don't memorize this without knowing the pathway, and as you learn the pathways, they will come by itself. So let's move on. Now, metabolism sites. Where does metabolism occur? So one, it can occur in the mitochondria, for example, fatty acid. What did I do? Okay, for example, fatty acid oxidation, that's beta oxidation, occurs in the mitochondria. Acetyl CoA production, here, this is the acetyl CoA production here. So acetyl CoA from pyruvate to acetyl CoA, production of this occurs in the mitochondria. That's what they are saying. Then the next one is TCA cycle, of course, occurs in the mitochondria, and oxidative phosphorylation occurs in the mitochondria. Ketogenesis, making of ketone bodies, also occurs in the mitochondria. All these are high yield points. They like to test all of them randomly. So fatty acid, a lot of stuff about fatty acid occurs in the mitochondria. A lot of stuff for the fatty acid. So beta oxidation of fatty acids, mitochondria. Ketogenesis, ketone bodies, we use fatty acid to make it. Then look at acetyl CoA production. Acetyl CoA can be used to make fatty acid, right? Can be used to make fatty acid here. So it also occurs in the mitochondria. TCA cycle, we know that, right? It occurs in the mitochondria. So cytosol, glycolysis, HMP shunt, and synthesis of cholesterol, proteins like ribosomes and ribosomes in the RER and production of proteins in ribosomes and RER, they're all in the cytosol, and then fatty acids and nucleotides are all in the cytoplasm. And then those what occurs in both of them, heme synthesis. So heme synthesis and what's happening? I don't even know what I'm doing. Okay, so heme synthesis, and then urea cycle, gluconeogenesis, all occur in both of them. We'll see that later. This is the summary of all the pathways. I think I have talked about this in some way, but then I can talk about it again. So first, the central part of our metabolism is what? Breaking down of glucose, that is glycolysis. All here is glycolysis to TCA cycle. Then all other things are linked to it. So what I want you to know is that do not study all the parts separately. The only time you can say you understand biochemistry is when you can link all these parts of the pathways together, so that you have to know that when this happens, it can join this. When this happens, it can join this. The central part can also make this. This part can make this. This part can come and make this. This part can come and make this. and then from here it can come and make this. So that is what you want to, that's how you want to learn biochemistry. That's how we learn it. So let's do a brief overview of this. Now we have glucose here, as you can see. Let me just stop this. Okay, we have glucose here, as you can see. Glucose can be broken down through all these pathways. That's what they want us to know, and then they want us to know some of the enzymes that are involved in this part. So they numbered the enzymes for us. It's not hard. This thing is not hard over here. Now, because it's a pathway that you always have to be referring to. So glucose will be converted into pyruvate. This is what we want to make from the glucose, pyruvate. But in the process of making the pyruvate, we'll make some ATPs on the way. Now, the pyruvate will be converted into acetyl-CoA. Now, this acetyl-CoA will go into the Krebs cycle or the TCA cycle, and then go through citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. That is it. That is the TCA. That is glycolysis and TCA cycle. Now, here, oxaloacetate can be used to make, from number 16 here, can be used to make PEP. Then this PEP will now start rising and going up to make glucose. That's what gluconeogenesis. So you can see that the pathways are here going upwards. Those that are going upwards. That's why they wrote it. The pink one here is the gluconeogenesis. But then when we're breaking it down, it's glycolysis. So glycolysis goes down to make ATP. Right, glycolysis will go down into this cycle, then to make ATP. But then at the end of the day, we'll make oxaloacetate, which can go outside when we need glucose or when we are starving. Then we use that to make glucose. This is happening in the third state. We've eaten a lot, so we have to break down the glucose. Now we are starving, so we have to make glucose. That's gluconeogenesis. Now, next, we said that we see galactose over here, right? Galactose is another monosaccharide, because in the monosaccharides we said we have glucose, fructose, galactose. Galactose can also be broken down. So over here you will see that we have galactose broken down into what? Galactose 1-phosphate. Also we've added a phosphate to it, right? And who added the phosphate? What enzyme will add the phosphate? So anytime we add the phosphate, we use a kinase, right? So we'll have galactose kinase, right? But then we don't have a galactose kinase, so it's most likely going to be let me see what they wrote. It's most likely going to, okay, that, let me see, yeah, that's what we have. So that's it. It's a long time I heard about something like that, galactokinase. Good, so it works. So just the substrate plus whatever the function of the function it is doing will give us the name. So galactokinase is the one that is doing this. So galactokinase will break galactose, will convert galactose into galactose 1-phosphate. All it's doing is adding 1-phosphate to the first carbon. That's why it's called galactose first carbon phosphate, or galactose 1-phosphate. And then that will also be converted into glucose 1-phosphate. And this enzyme is the galactose 1-phosphate uridyl transferase over here, right? And then from here, the uridyl transferase will transfer what? Uridyl to it, right? Transfer UDP, that is UDP, uridine diphosphate to it, and then they can be used to make glycogen. That's what we need initially to make glycogen, and then after making glycogen, we can be adding more glucose to it to make a larger glycogen. So in making glycogen, we need the UDP glucose first as the central part, as it's like the center of precipitation, the first molecule to be formed, UDP glucose. Then I'll be adding more glucose to it to make longer glucose. Now, so this can also be linked to what this glycolysis and glycolysis pathway, so that it goes down, because we can convert this glucose 1-phosphate can be linked here. And now we can also see here that acetyl-CoA can also, as we come down, glucose can make acetyl-CoA, we can convert to malonyl-CoA. This malonyl-CoA will be converted into what fatty acid. It also has a process, a pathway, which this is not the complete pathway, but then we can see that. And also this triglycerol. Triglycerol is not anything hard. It's a lot of carbons like this, dash carbon. Yes, I always draw the carbon. There are a lot of carbons with glycerol head. Assuming this is glycerol head. So if this is the glycerol head, then that is it. So triglycerol has a lot of this with glycerol head. So a lot of fatty acid with glycerol head. So if we split this out, we'll get the glycerol head, and this glycerol head can be used to make what the DHAP that we saw on the way. So DHAP, dihydroxyacetone phosphate, and can also be used to make this back fructose 1,6 bisphosphate. So I want you to know that everything is linked up. Don't learn them in isolation. Now, acetyl-CoA can also be used to make acetyl-CoA. It's just a combination of two of them. So if you combine two acetyl-CoAs, you get acetyl-CoA, right? And then that acetyl-CoA can be used to make HMG-CoA. This HMG-CoA can be used to make what? Mevalonate, and used to make cholesterol. This is a cholesterol that blocks people's arteries and cause what? This is a cholesterol that blocks people's arteries and then cause atherosclerosis and co. So when we give statins, so you can see that now there's another link over here. So when we give statins, we block what? HMG-CoA reductase, so that we cannot make this mevalonate and we can make cholesterol. So all these things make a lot of sense. HMG-CoA can also be used to make what? Acetoacetate, which are the ketone bodies. Yeah. And here, what you want to understand here is the odd chain fatty acids, leucine, valine, leucine, isoleucine, right? Valine, isoleucine here, and I don't see the leucine there, but then this can also, and the methionine and threonine can also be used to make what? Propanol-CoA. These ones they call them VIL. VIL has a branch, right? Branch chain amino acid. The branch chain amino acid. So I just call them VIL. My mnemonic, VIL has a branch, and I have a mnemonic for the amino acid if you want me to say it quickly. So it's like a song. It's ProGAVPIL fierce water. So these are those that fear water, the hydrophobic amino acid, ProGAVPIL. I'll put a mnemonic down someday. ProGAVPIL fierce water. VIL has a branch, so VIL are the branch ones. Then his Lys are so basic. So histone, lysine, and arginine are basic amino acids. So and L ketones. So lysine and leucine are for making ketone bodies. So if you again, I'll make a video about this song and then put it down so that you can appreciate it. So let me sing it again. ProGAVPIL fierce water. These are mnemonic for ProGAVPIL. They are all amino acids. And then ProGAVPIL fierce water. VIL has a branch. VIL has a branch. They are the valine, isoleucine, and leucine. Then his Lys are so basic. These are the basic amino acids. L and L ketones, that's leucine and lysine they are used to make ketones. So if you know this song, it helps. Now let's move to the next thing. Did we finish here? Oh, we didn't talk about the urea cycle. So fumarate. So in the urea cycle, we'll come to this. It's also very high yield, but we'll treat that separately. When we break down proteins, we said they will be used. Will form. When we break down proteins, will form what? Ammonia, right? Will form ammonia. Then we combine that ammonia with carbon dioxide, right? Then we form what we are going to form is carbamoyl phosphate. Now this carbamoyl phosphate can be what? Added to ornithine. Carbamoyl phosphate plus ornithine will give us citrulline, right? And then the enzyme there is called the ornithine transcarbamylase, because this ornithine, and then we are transferring carbamoyl phosphate to it. So we call it ornithine transcarbamylase. That is it here. And then the enzyme that is here is called the carbamoyl phosphate synthase, because we are using this and this to synthesize or to make carbonyl phosphate. So we call it carbonyl, this enzyme 20 is called carbonyl phosphate synthase. Then we transfer this carbonyl phosphate to ornithine to make citrulline. So we call it what? We call this one ornithine transcarbamylase. This is the most common deficiency. The most common deficiency that they always talk about. Now, if this is deficient, what will happen? We'll have excess of the carbonyl phosphate. We'll have increase in what? Ammonia as well, right? We'll have excess of this. We'll have excess of ornithine as well, but we'll have decreased citrulline. So just take note of that. The question will always say we have decrease in citrulline. So just take note of that. And then if there's also deficient, we'll have urea production will not happen, right? And then you want to know that at the end of the day, this urea cycle is what will give away one fumarate. That fumarate is also linked to the TCA cycle. So everything is linked up. Everything is linked up. Let's go to the next page. Now, ATP production. This page wants us to know how many ATPs are made from each of the pathways. For example, if ATP is made in for example the liver and then the heart, we'll have 32 net ATP via the malate aspartate shuttle. We have a 30 ATP when it's made in the muscles. This is not high yield. And anaerobic glycolysis produces only 2 net ATPs. Anaerobic means that what? No oxygen is involved, therefore the electron transport chain will not work. But then aerobic glycolysis, aerobic metabolism or aerobic glycolysis will end up into the TCA cycle, into the electron transport chain. That is why we'll have more ATP. But the anaerobic will only end at the pyruvate, right? Then anaerobic is also the same as when we don't have any mitochondria. So cells that do not have any mitochondria, like the RBCs, will also end at anaerobic glycolysis level because they don't have any mitochondria. Even if they have mitochondria and there's no oxygen, it cannot work. And now there's oxygen but there's no mitochondria, so it's the same thing. So take note of that. Arsenic causes glycolysis to produce 0 net ATP. Yeah, so that's a fact. Activated carriers. So this part is just trying to tell us that there are certain molecules. These molecules carry a certain thing that can be given away. So for example, ATP has a lot of phosphates, right? So phosphoryl group, NADH, DPH, and FADH. This is vitamin B3 here, and this is vitamin B2, right? They carry electrons in the form of the H. That's why they give it off in the electron transport chain. And then we also have a CoA, and then the lipoamine, they carry the acyl group. This coenzyme A is vitamin B5. Is vitamin B5, the pantothenic acid. So take note. Biotin is carrying carbon, right? And tetrahydrofolate is carrying 1 carbon unit. Biotin is carrying carbon dioxide because biotin is used in, that is vitamin B7, right? B7 is biotin, and we said it was used in carboxylation reaction. Carboxylation, adding carbon to something. So that is it. Whilst vitamin B6 was used in decarboxylation to remove the carbon. The SAM, we saw this in the vitamin B12, where we saw that vitamin B12 will be holding the B12 will be holding what? It will be holding CH3, which is the methyl group, and then add it to the homocysteine, and then the two of them will combine and form what? Methionine, and then the methionine will be converted into SAM, and the SAM will give off the CH3. This CH3 originally was with the tetrahydrofolate. TPP, this thymine pyrophosphate, also holds aldehydes. Yeah, this is somehow a little bit not, they don't really ask these kind of questions. I've never met it before, but you just have to have a general idea about it. Universal electron acceptors. So electron acceptors, as we saw, if, for example, these people did not accept the electron, they will not be carrying it, right? So NAD plus, NADP plus, and then which are all from vitamin B3. I call them vitamin B3, whether they are from Methanol, just know that they are all vitamin B3. And then we also have the other one, the FAD plus, or FAD. That is it, FAD, the flavin adenine dinucleotide. It's also another electron carrier. And then, yeah, we can continue. So NADPH is used in the HMP shunt, and then that is used in anabolic process respiratory burst. We'll see this when we do chronic granulomatous disease. They have deficiency of certain enzymes, and then we'll not be able to have those respiratory bursts. And we also use it in the reduction of glutathione. If you know glutathione, it's one of the vitamins. It's made in the body naturally, and it's when it's reduced, it can be used to remove free radicals from the body. Now let's look at hexokinase versus glucokinase. You see, this is what I don't like about first aid. Okay, it just states the thing. So if you are someone who does not really have an overview or a basic knowledge of biochemistry before you come here, it makes things very difficult. Now, why would they just jump and go straight to hexokinase and glucokinase? They have to link it to something. Where do we see it, right? So this hexokinase and glucokinase, for example, when we have, let me just show you this. You have a cell like this. Okay, this is the nucleus of the cell, and now this cell has glucose coming here, circulating around it. These are glucose, right? Let's say these are glucose. The glucose wants to enter the cell. Now we have glucose transporters on the cell. If they are able to carry this glucose, into the cell, okay, into the cell. Now, let's say the glucose has come into the cell, right? Now, because this glucose has come into the cell, it can also go back out. We don't want it to go back out. We want to hold it inside. So how do we hold it inside? We have something called a policeman, that is the P. So the P, which is the phosphate, will be attached to the glucose. So we have this glucose with phosphate attached to them. So glucose plus phosphate. So you have glucose 6-phosphate. You understand? So as soon as glucose enters the cell, we don't want it to leak back. So phosphate must be attached to them so that they don't hold that. The phosphate is P, right? So I just term the policeman. As soon as they enter the cell, the policeman catches them. Now, who makes the policeman to catch them? It is these two enzymes here, the hexokinase and then the glucokinase. So as soon as glucose enters, glucokinase adds a phosphate. We said all kinases. You see the kinase here? They've written kinase here, right? All kinases, what do? They add phosphate to something. So glucokinase will add phosphate to this. Hexokinase will also add phosphate to it. Now, what happens is that glucokinase, you realize that it is generally in the liver and then the pancreas, but hexokinase is in all other tissues. So basically, glucokinase is in the pancreas and liver only. Hexokinase is in all tissues, but they are all performing the same function. So just take note, they are adding a phosphate to glucose when it enters into the cell to become glucose 6-phosphate, like that. Now, let's look at the difference between them. What you want to know is that the location. You can see that they've written the location here. So glucokinase is in the liver, L for L, glucose, and then B in the pancreas. Hexokinase is in all other tissues except pancreas and the beta cells of the pancreas, except liver and beta cells of the pancreas. KM. Now, KM and then Vmax. you want to also know this. So Vmax is the maximum velocity of a reaction. That, for example, if hexokinase, which are enzymes, they are enzymes, right? So they'll need to speed up the reaction. So the maximum velocity of the reaction when the hexokinase is working would be what? That's a Vmax. Then the substrate concentration, let's say the substrate will be the glucose. What they are working on, that's the glucose. So the substrate concentration also is determined by the Km, right? So Vmax and Km are something that we have to appreciate. Now, they have a different Vmax and different Kms. So for me, hexokinase has low, and glucokinase has high. So what I just do, my mnemonic is low, like this, low. So hexokinase has low Km, low Vmax. I'm done. That's all my mnemonic for it. So you have to know this. Just know it. Low Km, low Vmax. Okay, just know this, that hexokinase has low Km and low Vmax, both of them. Low, hexo, low, and this hello, just like you're saying hello to somebody, and the opposite applies to the glucokinase. Okay, now when we say low Km and low Vmax, what does it mean? What does that mean? So when we say something has low Km, okay, it means that it has high affinity. So low Km means that high affinity, and then when we say it has low Vmax, it means that it has low capacity to work. So for example, if you look here, let's read here. They say hexokinase sequesters glucose in tissues where it is used even when glucose concentrations are very low, right? So when glucose concentrations are very low, hexokinase can still work, right? Glucose concentrations are very low, but then glucokinase can only work when we have high concentration of glucose, so that when we are hungry, certain cells will still have to take up glucose, and in those cells it's hexokinase that would most likely work. But then if you take the pancreas and the liver, if you're hungry, you don't want them to work, right? Because if they work, they'll release more insulin. You don't want that. Therefore, glucokinase can only work when we are in the fed state, that's when we have high concentration of glucose. So that's why they have a very low affinity for substrate. For example, glucose, for example, that's why they have high Km. Okay, because high Km means that they need high substrate, because we said Km represents the substrate concentration. So they need more substrate before they can work. They need more glucose in the body. They need you to have just eaten and have more glucose before they can work. That's why we call them high. That's why we say they are high Km. That is high substrate level. So Km can also be the substrate. Okay, Km represents the substrate concentration. So substrate concentration. When we say substrate, it may sound like somebody say, what is a substrate? A substrate is anything that we work on. I call it, example, food. If I'm eating food, the food is my substrate, right? So, and if an enzyme is working on anything, that is its substrate. So that's a concentration of the Km is the concentration of the substrate. So glucokinase will only work when the substrate levels are very high, or example, when the Km is very high. That's when glucose levels will work. So when you just eat, it means that you have a lot of glucose. Therefore, that's when glucokinase work. But then if we are fasting, then hexokinase will be the one working, because even when you have little glucose, hexokinase can still work. That's why we call it low. So at low substrate concentration, hexokinase can work. Let's look at the next one. So if you are still watching this video, you want to subscribe to this channel so that we can bring you more videos. We need more viewers. We need YouTube. The more people subscribe, the more YouTube suggest our video to more viewers. So please subscribe. And then when you comment, it means that our channel is interactive, and then YouTube will use that algorithm to suggest our video to more viewers. So thank you for subscribing. Thank you for commenting. Now, glycolysis regulation. So glycolysis regulation. So we saw earlier that glycolysis was the breakdown of glucose into pyruvate finally. Now, there are different steps in the glycolysis pathway. We need some of them, like some of them are highly regulated. We need those ones, but we don't need like all to understand like all of them into detail, because most of them are not high yield. The high yield ones are those that are highly regulated. So the first one is the hexokinase and glucokinase, the very first step in how do we call it, in the glycolysis pathway. So these pathways are usually irreversible in the sense that they need, if for example I'm doing glycolysis and I'm breaking down glucose, and then if I'm going downwards, okay, from glucose to pyruvate, if I have to go backwards to do gluconeogenesis, when I reach these enzymes, these particular enzymes that are listed over here, when I reach this enzyme, I will need a different enzyme to bypass them. That's why they said they are highly regulated. They can only work in one direction. They cannot be reversible. It's like in some of the pathways, you see that we have a reversible, so it means that the same enzyme can do gluconeogenesis and also can do glycolysis. The same enzyme can do opposite direction, but in these regulated ones, they can only do one direction, which is downward. So the first one is the glucokinase and hexokinase. What they do is that they convert glucose into glucose 6-phosphate. The next one is a fructose 6-phosphate. Fructose 6-phosphate being converted by the phosphofructokinase 1, the PFK1. That's the rate limiting enzyme for glycolysis. So PFK1 converting fructose 6-phosphate into fructose 1,6-bisphosphate, and then these ones require ATP, or these two require ATP. These ones produce ATP. The next one is the phosphoglycerate kinase. And so that is the one that makes the first ATP that we make, and then this is the second ATP that we make. So 1,3-BPG being converted into 3-BPG using phosphoglycerate kinase. Here we make ATP, and then here also we make ATP. I think in the next video we have to really take time to go through glycolysis, because it looks like they are ahead of us. The first aid is already ahead of us in the whole pathway, which that's why I don't like the first aid. You need to have a general knowledge before you do the first aid. But yes, if you are following carefully, you can pause the video and look at, and then have that page close to your page 74, so that you can really look at it. Now, regulation by fructose 2,6-bisphosphate. So here, what they want us to know is that fructose 2,6-bisphosphate and phosphofructose kinase 2 are the same bifunctional enzymes whose function is reversible by phosphorylation by protein kinase A. So if you look here, you will see that gluconeogenesis here, okay, and then here it's like we are using fructose 6-phosphate to make fructose 2,6-bisphosphate. Now see, what's the difference between these two? This one has only one phosphate on the 6th carbon, but when we come here, we are adding another phosphate on the 2nd carbon. That's why we call it 2-phosphate. That's bisphosphate, right? And then the enzyme that does that is PFK2. So PFK2 causes this, right? Now, this thing that has been produced by PFK2, that's phosphofructokinase 2, okay, can stimulate phosphofructokinase 1, which is in the glycolysis pathway. So in the glycolysis pathway, you realize that, let's say this is it. In the glycolysis pathway, where is the page? If you open to page 74, you realize that we have glucose being converted into glucose 6-phosphate. Then the next is what? Glucose 6-phosphate being converted into fructose 6-phosphate. Now, fructose 6-phosphate will be converted into fructose 1,6-bisphosphate, which is here. Fructose 1,6-bisphosphate. So fructose 6-phosphate being converted into fructose 1,6-bisphosphate. And then they want us to know that the enzyme that converts fructose 6-phosphate to fructose 1,6-bisphosphate is phosphofructokinase 1. Now, part of this fructose 6-phosphate can also be used to make phosphofructokinase 2. Then this phosphofructokinase 2 can stimulate the PFK1 to increase glycolysis. It makes sense. To increase glycolysis. Therefore, it's just like this is, you have this. For example, you have a fatty fish, a fish that has a lot of oil in it, okay? And then that fish can be used to make food. But then we are removing the fat, part of the fat from the fish to fry itself to make the food fast. I think this is a simple analogy. So you have fat, you have a lot of fat in a certain fish. Now, that fish can be used to make a meal. We need the meal to be made fast. We don't have oil. So what do? We remove part of the fat and then use it to stimulate or to fry it, and then to increase the cooking process. So fructose 6-phosphate is being converted into fructose 2,6-bisphosphate so that it can increase this reaction. It can stimulate the PFK1. Yeah. Now, in the fasting state, what happens? We make glucagon. So the alpha cells of the pancreas, the islets of Langerhans of the pancreas, will make glucagon. And so in the fasting state, we make a lot of glucagon. Glucagon can also stimulate what? Cyclic AMP. This one is very important. Glucagon increases cyclic AMP. If you don't understand this, you have to look at it from endocrine. Endocrine, we have all the mnemonics there, and glucagon is one of the things that uses the cyclic AMP pathway. Cyclic AMP will increase protein synthesis. And that protein is the protein kinase A, right? So protein kinase A, because cyclic AMP can stimulate the nucleus to make proteins, like to release nucleotides to make proteins. These proteins are the enzymes, and the example of one of the enzymes is the protein kinase A. Now, protein kinase A will lead to increase what? It's a kinase, so it adds phosphate. It increases FB pace, right? Which is here, this one, FB pace, and then that will and then also decreases PFK2. So meaning that in the fasting state, we don't want to break down, like to do a lot of glycolysis. We don't want to break down too much glucose. Otherwise, we'll go hypoglycemic. We don't want to do that. So in the fasting state, we are making a lot of this. This one here that they've mentioned here, fructose bisphosphatase. The fructose bisphosphatase is going to inhibit this whole thing. It's going to inhibit this so that we don't break down a lot of glucose. Yeah. In the fed state, we'll have more insulin. Insulin will downregulate cyclic AMP, and then that will lead to decrease in the protein kinase, and then that would lead to decrease in this thing over here. That's the FB pace, and then that will increase this one to increase this one, and then we'll have we'll have more glycolysis, but then we'll have less gluconeogenesis. So fasting state will slow the whole thing down. In the fed state, everything will increase. So that should make sense. Fasting state, you don't want to break down a lot of glucose. In the fed state, you want to break down a lot of glucose. So that is it. Pyruvate dehydrogenase complex. So in the previous video, we talked a little bit about the pyruvate dehydrogenase complex, whereby we said we have a lot of dehydrogenases, and one of them is the pyruvate dehydrogenase complex. So in the mitochondrial enzyme complex link with linking glycolysis and TCA cycle, and then differentially regulated in fed or fasting states reaction. So we have the pyruvate plus NAD plus coenzyme A plus giving us acetyl CoA plus plus carbon dioxide plus NAD. So you just have to know that the pyruvate dehydrogenase complex has all these things in it. So we have the thymine pyrophosphate, we have the lipoic acid, we have the coenzyme A, which is the vitamin B5. Sometimes they just ask the vitamin B5. They don't mention the coenzyme A. Sometimes they ask coenzyme A, and then they want you to know what it does. And then we have FAD as part of vitamin B2, and then we have B3 in it, niacin. So we have B1, B2, B3, B5, and lipoic acid, or lipoic acid, or some people just say it's a lipid, lipoic acid. Yeah, so B1, so the only thing that is not here is B4, right? So B1, B2, B3, B5, and lipoic acid, they make up the pyruvate dehydrogenase complex. It's really easy. Yeah, and then it's activated by increase in that's low energy state, or increase. So increase NAD plus. Anytime you see more NAD plus, that's a low energy state. If you see more, this one is high energy state. So this is increased by low energy state, and it's also another low energy state is here, right, which is ADP, and also another low energy state here, which is calcium. So calcium will also stimulate the pyruvate dehydrogenase complex. Yeah, the complex is similar to the alpha ketoglutarate dehydrogenase complex that we saw in the TCA cycle. We see alpha ketoglutarate over there. So at the end, we have to really take glycolysis and treat it separately, and we have to take the TCA cycle and treat it also separately. So I'll work on that video also and bring it to you. So please subscribe so that you can get that video also. So this pyruvate dehydrogenase complex is similar to the alpha ketoglutarate dehydrogenase complex. So they are similar, the coenzymes and then are the same. It converts the alpha ketoglutarate into succinyl CoA in the TCA cycle. Yeah. So here the mnemonic here is the lovely coenzymes are for nerd. Lovely coenzymes for nerd. Okay. Okay, now let's look at the next page. Now, this is the next page. So the pyruvate dehydrogenase complex deficiency causes a buildup of pyruvate that gets shunted to lactic acid. If you want me to take you back, this is what they are talking about. You know, when we have pyruvate can be converted into what? Acetyl CoA, as we said before. The acetyl CoA will go into the TCA cycle. Now, what enzyme does that is the pyruvate dehydrogenase complex, right? Pyruvate dehydrogenase, that does that. So when it is deficient, now we'll have a lot of this pyruvate that will build up, right? If pyruvate is building up, what will happen? It will be converted into what? Lactic acid, most likely into lactic acid. And that lactic acid can cause pain, right? Muscle pain and so on and so forth. And then they are saying that this is an autosomal, and it's an X-linked. It's X-linked, most likely recessive. To be X-linked, so it's X-linked. So the conversion of the pyruvate to lactic acid is done by this enzyme lactate dehydrogenase, right? So pyruvate will be converted into lactic acid by lactate dehydrogenase due to the deficiency of pyruvate dehydrogenase. The same way if somebody lacks mitochondria, they will also have pyruvate being converted into lactic to lactic acid, or lactate. Yeah, and this is this is called a Cori cycle. Findings of the pyruvate dehydrogenase complex deficiency: we have neurologic deficit or the neurologic defect, we have lactic acidosis, we have increase in serum alanine starting in infancy, because this is a fate of pyruvate. After we make glucose, pyruvate can be converted. After we convert glucose into pyruvate can be converted to all these things. Okay, now if we lack the pyruvate dehydrogenase complex, it means that we'll have more of the pyruvate to be converted to all this stuff, and then one of them is the lactic acid. Correct. Now also it can also be converted into alanine, right? So why they say increased serum alanine starting from infancy, and it never goes away. If we lack this, we'll have more of them going to alanine. That's ALT, right? Serum alanine, ALT. Yeah, so ALT is the one that converts, so that's alanine transferase, or alanine amino transferase, ALT, AST, ALT. That's it. Treatment: we have to increase intake of ketogenic nutrients. That's high fat content, or lysine and leucine. If you remember my mnemonic, what did I say? ProGAVPIL fierce water, Vil has a branch, his Lys are so basic, L and L ketones, that is it, ketogenic, L and L ketones. So this L and L, lysine and leucine, make ketones. So these are the ketogenic amino acids. So we give them more ketogenic amino acids, or we give them high fat content, so that they don't get a lot of lactic acid. Because if we give them glucose will make a lot of pyruvate. The pyruvate cannot be broken down into, or cannot be converted into acetyl-CoA. Therefore, it will be converted into lactic acid. So these children, when they lack this pyruvate dehydrogenase complex, they would be having lactic acidosis from infancy, right? So we don't give them glucose diet, we rather give them ketone diet. Take note of that, very, very high yield. We give them keto diet, and then they'll always ask you which amino acid should be included in their diet, and it should be either L and L ketones. Just sing the song L and L ketones, and then you'll get it. Okay, so the answer will always be lysine or leucine. Nobody will ask you high fat diet, because everybody knows that keto diets have fat. Okay, now functions of different pyruvate metabolic pathways. Functions of different pyruvate metabolic pathways. So here they are. They are just talking about the fate of pyruvate. One is pyruvate can be used to make alanine. That's, yeah. And we use B6 over there. B6, we'll see that somewhere. And just as they mentioned here. Now, pyruvate, so for example, this ALT that is using pyruvate to make alanine is the one that we see when somebody has cardiac enzymes that are high, the cardiac enzyme, the liver enzymes. You see, when we have liver enzymes, for example, when someone has liver disease, we have ALT and AST, indicating that there's a liver damage, right? Another thing pyruvate can be used to make is the oxaloacetate. That is very simple, oxaloacetate. And also, pyruvate can be used to make acetyl-CoA that will go into Krebs cycle, and pyruvate can be used to make lactic acid, right? So if somebody is has increased pyruvate, these are the things that they can use the pyruvate for. This is a TCA cycle. Now, the TCA cycle, we saw that from gluconeogenesis, glucose was being used to make, so over here we have glucose here. Glucose was being used to make what? Pyruvate. Good. Now this pyruvate can be used to make what? Acetyl-CoA. Acetyl-CoA will go into the mitochondria, right? And then be used to make all these pathways. Now, in each of the pathways, we start with 2 carbons here. So you can see they've written 2C. That's 2 carbons. Then we combine this 4 carbon with it, which is the oxaloacetate, to make citrate, which is a 6 carbon. Right. And now the next thing is this 6 carbon will be converted into isocitrate. Right. And now isocitrate will be converted into alpha ketoglutarate, alpha ketoglutarate, which is a 5 carbon. Here we'll lose a carbon over here, carbon dioxide. Therefore, we have 5 more carbons left. Here is where we need the pyruvate dehydrogenase complex. Here it's called the isocitrate dehydrogenase. So this is one of the dehydrogenase. Take note of this NAD here. It's tested. Next is the alpha ketoglutarate being converted into succinyl-CoA, and now it's also losing 1 carbon, right? Therefore, we'll have 4 carbons left. So the enzyme here is the alpha ketoglutarate dehydrogenase. It's also a dehydrogenase, right? It's removing hydrogen. That's why we have this hydrogen here. Here is also dehydrogenase, so it's removing hydrogen from here. So we'll have this hydrogen here, right? And here also we make NAD. Here we make ATP. Take note of where we make the ATP, because this is highly tested. Okay, now this succinyl-CoA can be used to make heme. So you see how even glucose, something in glucose can be used to make heme. Sorry, I have to learn how to use this. Okay, I'll learn it. Everything is learnable, right? Okay. Yeah. So this succinyl-CoA can be used to make heme. So now you appreciate that glucose here can be used to make something here to form heme. So this succinyl-CoA can also be converted into succinate, and then we get a GTP from here. This GTP is very important. It's used in nucleotide synthesis. Now, succinate, which is still 4 carbons, can be used to make fumarate, which is still 4 carbons, fumarate to malate to back to oxaloacetate, and the cycle continues. The things that you want to remember here is that this NH3 here, 2. So this is 1, 2, 3, NADH. That's what I want to say, NADH. This 3 NADHs, you have to pay attention to them, because you won't like asking where they are found. Between conversion of what and what would you find an NH3? Usually it is alpha ketoglutarate to the succinyl-CoA, so KS, right, and MO. So I just picture it. I place 1 at the top, I place 1 here, and I place 1 at the bottom. So I just picture it. Now, the GTP, they also like to ask where it is made. It's made from succinyl-CoA to succinate. That's where we make the GTP. So they love to ask that also. So pay attention to this. Okay, pay attention to this cycle. Yeah, and they said the enzymes are irreversible. So the enzymes are irreversible, but then they can slow down. If we have excess of any of the substrates, they can slow down. As we said, when we are in the fed state, or if somebody takes alcohol, the person will have so much of the NADH, right? Because NADH means that we have excess NADH, means that we have a lot of energy. So all this thing will slow down, and we'll use the citrates to make other things like ketones and the rest. Yeah. Okay. So we can move on to the next one. Here, this page has some high yield information. So this is the electron transport chain. So after we've done all this TCA cycle, the NAD, the NADH, all these 3, and then the FADH, this one also, will now, they are carrying electrons. This H that they are holding are electrons, right? This H here are electrons. The H they are holding are electrons. So this H will now be transferred through the mitochondrial membrane, the inner mitochondrial membrane, right? And then they will be taken to this complex here. Finally, this is the ATP synthase to make the ATP, and this is known as the oxidative phosphorylation. These are the electron transport chain, and this is the oxidative phosphorylation, meaning that we are using oxygen to do phosphorylation of ADP to ATP. Okay, that's why I call it oxidative phosphorylation. This is a phosphorylation, adding phosphate to ADP to make ATP. That's phosphorylation by the presence of oxygen. That's when there's no oxygen, all this cannot occur. So now let's see. Now what you want to know here is that we have different complexes. Okay, some of them have names, some don't have other names. So from complex 1 up to complex 5. Now in here we have complex 1, okay, takes the NAD, okay, and then pumps the hydrogen. It pumps the hydrogen out, right? And then we are left with NAD plus. Now this hydrogen is going to be given to coenzyme Q here. So these hydrogens that are taken, that we get from here, will be passed on to coenzyme Q. Now complex 2, that's the succinate dehydrogenase. And then we saw this succinate dehydrogenase. Where is it? Here. It is right here. Succinate dehydrogenase. It should be either here or here. Let's see where it is. Okay, very, very important, because we have to pay attention to it. Yeah, here. So here, this one here, we have succinate SD, succinate dehydrogenase, right? Okay. Good. So succinate dehydrogenase over here is the same as what we have over here, and then they said it's in the mitochondrial membrane here. So we'll have the succinate dehydrogenase here. It's the same as what is over there. So just take note, okay? It's the enzyme that is in both TCA cycle and also in the electron transport chain. So succinate dehydrogenase will also take all the electrons from FADH2 and then pass it on to the same coenzyme Q, right? Then the coenzyme Q will give it into complex 3, right? Complex 3 will now use cytochrome C to pass it on to complex 4. Now complex 4 will now be the final electron acceptor, which will now give the electrons to, which will now use that how do you call it, it's not like they're not giving the electron directly, but let me show you exactly how it happens. Now anytime complex 1 gives off its NAD, the NADs are positively charged, right? They move from here and then they come here. So we'll have a lot of them, a lot of the NADs, a lot of the H pluses coming out here. A lot of the H pluses coming out here. A lot of them, because these are the H pluses, right? Now, this one will also do the same thing. But then when we have excess H plus here, you see that this will become positively charged relative to the inside of the mitochondria. And that electron gradient, or the potential differences, generate a certain force called the electromotive force. Electromotive force. So it is that electromotive force that we gather at the end of the day, and then that force is used to combine ADP and then the inorganic phosphate here, this phosphate, to make ATP. And then what makes the ATP finally is this man over here. That's the ATP synthase, or ATP synthetase, like that. So this is the mitochondrial matrix here, the inner mitochondria, and then the intermembrane space here. So that's what you want to know. The mitochondrial matrix is where we are pumping all these things into. The matrix is the more of the inner part of the mitochondria. That's why we are pumping all the H plus into to create that electromotive force as to generate to help us to synthesize the ATP. Yeah. So there are some things that you have to know. This Draco is something that is sometimes tested. You want to know that in I need to connect my charger. Sorry. Before this thing dies, because I don't like this. It's 1%. Gosh. Hurry. Good. We are back. Sorry. Now, so what we were talking about is that, sorry about that. So what we're talking about is that all these things are inhibitors of the electron transport chain. All these are inhibitors of electron transport chain. So you want to know each of them and how they inhibit, right? So we have metformin here, and then the rotin 1. Rotin 1, the 1 over here means that it's 1. Rotin 1 is inhibiting complex 1. Then we have antimycin complex 3. And 3, people call it antrimycin. That's how I call it. It's here. Antrimycin. It's inhibiting complex 3. Antrimycin. And then we also have azides, and then we also have cyanide and carbon monoxide can inhibit complex 4. Oligomycin can inhibit complex 5. Right? So like that. And when they inhibit all these complexes, it means that we are not able to make enough ATP. Right, because they block the pathway, so we cannot make ATP. So in this session, you are not supposed to know exactly one, because I've not seen question that is asking you which one is complex 1, which one is complex 2, which one is complex 3. Yeah, but you may find questions that are asking you about, for example, how do we call it, rotin 1 blocking complex 1. It's Amboss that likes this kind of questions, but step does not really go after this thing, but just know the general overview, and then know that we have coupling and uncoupling substances. For example, these things over here are the uncoupling reactants. So coupling means that when we say something is coupling, like they join together to form ATP, for example. So if at the end of this reaction ATP is made, then we call it a coupling reaction. But then if at the end of the reaction ATP is not made, then it means that it is an uncoupling. It's not coupling, it's an uncoupling reaction. So substances that can cause an uncoupling reaction are these two here, the dinitrophenol aspirin overdose. What will happen in an uncoupling reaction is that you will have all the H plus going into the the mitochondrial matrix, all right. The electromotive force will be generated, all right. By the end of the day, ATP will not be made, so heat will be produced. Like some amount of heat will be produced that can destroy the organism, that can cause death in some animals. That is why when you give so much aspirin to somebody, they can have increased body temperature, because it's causing an uncoupling reaction. Uncoupling reactions produce heat. So these uncoupling reactions can occur in the brown fat that is at the back of babies. It occurs in cold-blooded animals like snails. Snails can live in the cold, and then they'll just be hiding there, and then their metabolism is very low, but they can produce heat inside them like that. So that's uncoupling reaction. So increased permeability of membrane, causing decrease in the protein gradient and increase in oxygen consumption. And then ATP synthesis stops, but the electron transport continues. This produces heat. As simple as that. So it's sometimes useful, sometimes not useful. So yeah, like that. And ATP synthase inhibitors. So directly inhibits mitochondrial ATP synthase, causing an increase in the protein gradient. No ATP is produced because electron transport stops. Yeah, of course, if you block this ATP synthase, no ATP will produce. And then we understand like uncoupling. So uncoupling make the reaction occur, but then at the end of the day ATP is not produced. It's therefore heat will be generated. Let's look at the last one here. Let's see if let's look at this, and then this will be another topic for another, the pentose pathway. Gluconeogenesis irreversible enzymes. So in gluconeogenesis, just as we saw in the rate limiting enzymes for the glycolysis, that we said those ones are irreversible, in the gluconeogenesis as well there are irreversible enzymes. So these are the pyruvate carboxylase, the PEP carboxykinase, or we'll just call it I call it PEP, that is phosphoenol pyruvate carboxykinase, and also the fructose 1,6-bisphosphate 1, and then glucose 6-phosphatase. So these ones are irreversible when we are converting, for example, we are going backwards instead backwards to make glucose, and yeah, so we can look at that also. And so thank you for watching this video. Please subscribe and share to invite more people to this channel, so that we can all interact together. If you don't really understand anything that we talked about, and if you also have any suggestion or anything that didn't go well in the video and then you want to add it, we are all educating ourselves, and we are all still learning. So please take time to write your comments, and I'll be glad to also learn from you. And thank you so much for joining me today, and please enjoy your studies. Have a great day and a wonderful studies. Thank you. Bye-bye.